Hi friends, this video is about the variation in the protein structure. So we know that the protein structure is a complicated structure that is 3D structure which may exist in the primary, secondary and tertiary and quaternary structure. So primary structure are the sequence of DNA, example insulin which have 21 amino acid and B chain have 30 amino acids. For example, there is another example that this oxygen transport protein hemoglobin consists of four polypeptide chain, two alpha and two beta, identical alpha and beta chain. Sickle cell anemia, a single amino acid substitution in the beta chain causes a change in the structure of the entire protein when the amino acid glutamic acid is replaced by the valine in the beta chain. The polypeptide folds into a slightly different shape. This example tells about the disorder of the primary structure, and this disease is called as sickle cell anemia or sickle cell disease. Secondary structure is a regular structure as in the interaction between the neighboring or nearby amino acid as these polypeptides start to fold into its function 3D form. So interaction between the H bond and between the local group of amino acid. There is a most common form of the secondary structure are the alpha helix and the beta pleated sheet. So next one is a tertiary structure. It is a 3D structure, and interaction between the R group, that is alkyl group of the amino acid. Interaction may be in between the H bonding, ionic bonding, dipole interaction, hydrophobic bond, and then it contribute to the tertiary structure. Covalent bond formation and the disulfide bond is also found. The fourth one is the quaternary structure, in which the multiple polypeptide chain, also known as the subunits, come together. They give rise to the protein and its quaternary structure. And the structure of the protein are folded into endoplasmic reticulum, which is correctly folded in a specific 3D shape. In order to function correctly, the unfolded or misfolded protein contribute to the pathology of many diseases. The chaperones and the chaperonine regulate the correct folding of the protein. Any change in the nucleotide sequence of the DNA alter to genetic codons and thereby the alter of the amino acid sequence of the primary structure of the protein is affected by the genetic mutation, which is maybe of the two types, that is mutation, synonymous mutation, also known as the silent mutation, and the non-synonymous, which is very important for study. First one is the synonymous mutation, in which the changes in the codon but no change in the amino acid. Example, alanine, leucine and the valine, in which the C is replaced by the A and still its amino acid valine. Non-synonymous is of the sub two types, that is missense mutation and the nonsense mutation. Missense mutation resulting the another amino acid but not the terminal codon, and nonsense mutation leads to a terminal codon, that is stop codon, and protein stop to synthesize another translation stops and the protein synthesis is stopped. Each of the sense codon can mutate to 9 other codon and by means of single nucleotide substitution. For example, a CCU proline can experience 6 non-synonymous and the 3 synonymous mutation. So there are 549 possible nucleotide substitution. So almost 70% of all possible nucleotide changes at the third position are synonymous mutation, and the second and first position of the codon are non-synonymous. Vast majority of the nucleotide changes in the first position, that is 96%. It is said to denaturation that the means when what is denaturation means, it is a change in the or the protein loses its higher order structure but not its primary structure. It is said to be denatured. It is non-functional factors. There are several forces which affects the this protein structure, that is electrostatic forces, H bonding, hydrophobic forces and disulfide bonds. So all about the variation in the protein structure. Keep watching.